Let's get started officially. So it's a great pleasure for me to introduce Deanna Benson. Deanna will be speaking in our new cyber series of lectures for the Friedman Brain Institute, which will continue through the fall and even next winter and spring. It gives an opportunity for the neuroscience community here at Mount Sinai to hear from some of our own faculty, which is something we rarely do. So it's a little bit of a silver lining of the time of COVID. That we were prompted to do this. It's been very popular and successful, and I'm really very pleased to be continuing it. So today's speaker, Deanna Benson, has been a longtime member of the Mount Sinai faculty. She's a leader in neuroscience nationally. Her laboratory has focused on different aspects of developmental neurobiology, how neurons form circuits and maintain them. And for the last, I think maybe 5 or 6 years, Deanna has also been director of the Microscopy Core at Mount Sinai and has brought many technical innovations for that core, has done a spectacular job. So Deanna, thank you again for doing this. And it's great to have you speak to us. Thank you.
Thanks, Eric. I'm gonna share my screen. Okay, and so can everybody see that?
Yes, looks good.
Okay, and so the title of my talk, I debated for a long time about coming up with a title that covered basically every single thing that I'd ever done. And so it's construction, flexibility, and susceptibility of adaptable circuits. But the title that I threw beneath it, the MacGyver principle as applied to synapses, is the title I actually wanted to use. But I wasn't sure if those of you who didn't grow up in the 80s would know who MacGyver was. And so I'll get into what that means in a minute. Let's see, laser, there we go. So the basic strategies in my lab have been kind of twofold. The first is from the bottom up. And that's the idea behind that is that we'll never really understand things until we see them forming from the beginning. This is kind of the driving force, I think, behind a lot of developmental biology. The second is top down and is something we've been focused on more recently, and that is that gene mutations that are identified in human brain disorders can be leveraged to define both what's pathological and what's normal. Both texts have revealed kind of this underlying theme of adaptability. And so we started this several years ago when Wendong Zhang was in the lab. This is a photomicrograph from Alice Elstey. But the idea behind these first experiments were that the synapse is basically an adhesive junction, like any ordinary epithelial junction. And this EM shows it very clearly that even at a first forming, developing synapse, you can see that you have these opposed membranes, you have this gray material in the cleft in between, and a cluster of presynaptic vesicles. But in the inherent structure of this particular part, it looks like an adherens junction. And adherens junctions are, they join two cells by having adhesion molecules that are embedded in an actin cytoskeleton. And based on this, if you depolymerize the actin, you should disassemble the junction. And so the first series of studies that we did were kind of asking very simple questions, whether or not actin really condensed its sites of contact, as you would predict if this is an adherens junction kind of a structure. As you all know, F-actin nicely concentrates in growth cones. You can also see patches along developing axons. You see lots and lots of actin in the somatodendritic domain. So this is a cell body and developing neurites. It looks incredibly bizarre, but these big chunks of F-actin, in fact, aren't abnormal. This is perfectly normal, and you get these big concentrations of F-actin in developing glutamatergic neurons. We think it's probably a resource needed for the rapidly expanding dendritic and axonal arbors. And then as synapses are first forming, this is just synapsin labeling the presynaptic terminals in green. You can see these condensations of F-actin that occur at the sites of synaptogenesis. So as synapses are first forming, we do have concentrations of F-actin. The F-actin stays enriched at least postsynaptically throughout life, and you can easily see it in this tissue section stained for phalloidin in the hippocampus. You get these large concentrations of F-actin marking dendritic spines. So actin condenses at the sites of contact. The next thing we did was ask whether or not actin was actually necessary for synapse adhesion. So the upper panels show our control and the lower panels show neurons that have been exposed for 4 hours to latrunculin A, which is a actin depolymerizing agent. And in the control samples, you see these nice these are just young neurons, just first forming synapses. And you can see these nice presynaptic terminals and postsynaptic sites. Here it's shown, even here you already have a PSD. And you get these clusters of synaptic vesicles concentrating along the somatodendritic domain. 4 hours after latrunculin A, what you see are these huge numbers of vesicles in both axons and dendrites. The cell body looks actually astonishingly normal. And in the somatodendritic domain, you see a distribution of all these nice clustered vesicles and now become spread throughout the arbor. So you no longer have polarity of the vesicles. They're distributed everywhere and you have nothing at all that looks like a synapse. So synapses are absolutely destroyed by this treatment. And so we were really excited by this. So it's like, okay, synapses basically follow the rule of an adherens junction until we looked at older neurons. And so these are just neurons grown in culture for a couple of weeks. Actually, these are 18-day-old neurons. And you can see the F-actin is an elaborate, more elaborate structure than I showed you before. And you have lots and lots of presynaptic green synapsin-filled terminals. This is an EM of a similar prep. If you depolymerize the actin, now it takes longer. You have to depolymerize for 24 hours. And you can still see occasionally these little pink F-actin patches. But the bottom line is the synaptic vesicle clusters remain. You keep synapses. And if you look by EM, you see still a really nice pre to postsynaptic structure. So F-actin is absolutely dispensable for synapse maintenance. So why is this possible, or how is this possible? In these intervening weeks, we've slowly accumulated the PSD proteins, these scaffolding proteins like Shank and Homer and PSD95 that together make this dense PSD. So we have this amazing PSD network that now is sufficient to hold at least a sufficient number of pre to postsynaptic adhesion proteins embedded in that matrix so that when you get rid of the actin, you still maintain that pre to postsynaptic structure. But the synapse is not the same. We're missing actin. And this is where the MacGyver principle first comes in. And so this is for the benefit of those of you who didn't grow up in the 80s. This guy is MacGyver. He's got a nice mullet, but that's why he's not why he's famous. And that's not why I'm using him here. He was like kind of the way to describe MacGyver is he's sort of a mashup of a peacenik, a special ops kind of dude, and a mythbuster. And so he was really famous for making or repairing an object in an improvised or inventive way, making use of whatever items are at hand. And he was so famous for this, and he invented so many like random weird things in, tense situations. The Oxford Dictionaries has now incorporated this as a verb. Here he's making, I think this is the episode where he made a hot air balloon out of a refrigerator, some welder's parts, and a parachute. And so in our case, the synapse, now MacGyver's his inventions always worked. But in biology, I think the inventions don't always work so well. And so they either work temporarily or they work differently. And so we have generated a synapse that is functional. So we have recorded from these neurons, they're functional, but in fact, they're shaped differently. And so we all know now that this is a di-eye-leveled neuron in culture. And you can see we have nice dendritic protrusions that have these nice puffy ends. These are like mushroom-shaped spines. You depolymerize the actin. In fact, while the synapse is still attached, it's as though you deflated the spine. It's pointy. And that spine is abnormal. And that shape is relevant. And so we know that the shape is relevant because bigger synapses are stronger synapses. Lots and lots of evidence shows that these big mushroom-shaped spines have lots and lots of, have more AMPA receptors. The shape itself imparts information. We know with long-term potentiation that spines expand, and that expansion is necessary to retain stronger responses. There's a skewed shape distribution in neurodevelopmental disorders. This was first described, I believe, by Dominic Purpura in the 70s, where he was looking at pathological samples from children who'd had intellectual disability, and he noticed that shapes of dendritic protrusions tended to be pointy and thin. And the loss of small, thin spines in aging correlates really well with cognitive impairment and dementia. This is work done by Mark Baxter and John Morrison in non-human primates, and now there's correlative work in aging humans. But this loss selectively of thin spines in aging seems to be really important to being able to generate new memories. Additionally, actin seems to be really critical to imparting activity-mediated signatures, for lack of a better word. And a simple example of this is shown here in an experiment that Wendong did, but this is normal actin in the presynaptic terminals like you saw previously. If you stimulate briefly, a hard stimulus, this is KCL for 5 minutes, you can see this increase in actin, an increase in actin intensity. So this is phalloidin, just it appears more intense. If you then try the same latrunculin A experiment on this group, you can see that while you depolymerize the actin, normally you depolymerize the actin and most of it goes away, and then the synapses are retained. Here, you depolymerize the actin and more of that F-actin is retained, and most of that F-actin that's retained is retained at synaptic sites. So there's something about activity and strong activity that helps impart information. And this is important in particular during development with this notion of synapses that fire together, wire together. Actin is also really important for supporting spine dynamics. Spine dynamics are important just for generating normal synapse function. It's required for long-term potentiation. Spines are most dynamic during development, and they can be silenced by anesthesia. So this is something that Andrew Mattis showed many, many years ago, but this is the normal kind of developmental wiggling. And you can see with isoflurane that they cease. They just cease moving at all. And this is an interaction that was discovered in 1999, but it's also seen with hypothermia, and it comes up now when people are talking about possible impacts of anesthetics or even cooling that might happen during surgery and the effects that they might have short-term and long-term on spines and memory. So how do we generate actin at synapses? And so, these studies have been around for a long time. And so it seems like we should know all this, but the short story is that we don't. And so we know quite a bit about ARP23. So ARP23 is shown that generates branched actin filaments. And the ARP23 complex is made of about 7 different proteins working together. They form a base here on a pre-existing actin filament, and they work coordinately with a factor, they often like WAVE or WASP, in order to generate actin polymerization at the membrane. And so cell adhesion molecules typically work coordinately with something like WAVE to promote polymerization at the membrane. And they're recruited by RAC, and there's been many, many studies in neurons on this system of generating actin at spines. But in fact, there's an entire family that's relatively ignored relative to ARP23. And so there's a family of about 15 Formin complex proteins, as well as what WH2 domain proteins, which generate straight filaments. And Formins form a dimeric complex that sits at the very tip of F-actin fibers, adding on to the barbed end of F-actin and then pulling up the Formin molecule behind it. This large group of there are about 20 different nucleators that are expressed in the central nervous system, many of which are known to be in neurons, and most of which we know virtually nothing about. What we do know suggests that the shapes that we have reflect balances between actin nucleators. So spines that are predominant, or if you, protrusions that are dominated by ARP2/3 complex and ARP2/3 associated structures are more puffy and mushroom shaped, whereas those that are dominated by formins tend to be thin and filopodial-like. And knockout or knockdown preparations tend to skew the population one way or the other. But in truth, all of these populations have mixed filaments. So there are mixed straight and branched filaments in all populations of protrusions, and mixed filament types are really important for specifying particular types of endo or exocytosis. So it's too simplistic to just think everything is ARP2/3 in a spine and everything is Formin in a filopodium. So the mechanisms mediating assembly to impart stable structures in neurons are very, very poorly understood. So how do multiple nucleators coordinately regulate synapses? This is a question that is being led by Abhishek Sahasrabudhe in the lab, and he's been working with a slew of students. Here's Fatima and Chanel. I'll be showing you some of their data along with Aaron. To embark on this project, we wanted to be able to see what do each of these nucleators do in the context of synapses. And in order to do this, we had to build back a system that permits us to examine multiple nucleators in a medium throughput kind of a fashion. And so to do this, he has been injecting DNA into the cerebral cortex and doing electroporation at E16. This is a strategy that enables him to label selectively layer 2/3 neurons, which is going to decrease the complexity of our sample. He takes these filled neurons and he tags them or immunostains them with Homer, which is it's a postsynaptic scaffolding protein that is evenly distributed throughout the PSD, and VGLUT1, which is a tag for excitatory terminals. And this enables him to look at innervated synaptic protrusions. So he's been developing this strategy using Emaris, in which he's been segmenting dendrites and spines and classifying them, and then taking the Homer and VGLUT spots and determining how close, and identifying those that are close to the dendritic spines, and then masking them to look at this coordinated action between them. So in order to test whether or not this strategy of tagging, labeling, and going through image analysis is sensitive enough in order to detect differences in actin-mediated effects on protrusions, he initially did a study where he stimulated the neurons with CLTP. This is a chemical stimulation paradigm that induces long-term potentiation in a culture, and you can increase spine volume. And also, encouragingly, if you inhibit ARP2/3, you abolish this change in spine enlargement. And not surprisingly, mushroom and stubby spines are most affected. So if this color here refers to the mushroom spines, you can see with LTP, you get an expansion, and inhibiting ARP2/3, you get in a subtraction or retraction of this category. Additionally, he's discovered that dynamic actin is required to restrain PSDs. So normally chemical LTP or LTP generally doesn't change Homer volume in controls. And so this is something that has to do with timing. Over time, once you have spine expansion, the PSD will also expand to fit. But over these short periods of time, there isn't time yet for the PSD to adjust. And in typical samples, the PSD size does not change with chemical LTP over the time frame we're looking at. But in fact, even under control conditions, if you inhibit formins, the actual PSD begins to expand. And on stimulation, both either inhibiting ARC2/3 or inhibiting formins leads to this expansion of the PSD. This expansion is matched, the postsynaptic expansion is matched by an expansion on the presynaptic side. And so what these data are saying is that you need constant dynamic actin in order to actually restrain these structures normally, which is also consistent with what we know these structures do in the context of constantly recycling receptors and stuff at the edges of PSDs. So the next thing we examined was you have 20 or so different formins. Each of these has to have a different job. It doesn't make sense that you would have a whole bunch of redundancy in the context of making the same kind of filaments over and over, and that it didn't matter who was recruited when. And so modest differences in each kind of formin are predicted to have very different jobs implemented in very particular domains. And consistent with this, Abhishek expressed Formin 2 GFP in neurons, and here it's shown in blue. You can see the Formin GFP forms either these patches along the actin cortex of the dendrite, or it invades just the neck of dendritic spines. This is a very restricted distribution. It's only at dendritic, the neck part of dendritic spines. Separately, there's been like one other report of where formins are in dendrites, and Formin like 2, the Gioni lab showed, is only at the tops of dendritic spines. So these two pieces of data together suggest to us that in fact, different formins will be recruited for different jobs at different sites. Additionally, this was a good backup to kind of let us use Formin 2 as our example for our screen to see, we would predict that eliminating Formin 2 is going to have some kind of a readable outcome in our cultured system. And so Abhishek, now in coordination with Tim Offelt's lab, we've built a library of all of these different formins, guide RNAs against all these different formins, coupled with Cas9, and the idea is we'll infect neurons or transduce neurons with lentivirus, and then monitor what the outcomes are in neurons missing particular formins. As a positive control, Abhishek first looked at Formin 2. And additionally, the reason to look at Formin 2 is that Joe Gleason's lab showed that humans lacking Formin 2 have intellectual disability, and a mouse model lacking Formin 2 has an age-related cognitive decline. In mice expressing, in the neurons expressing a guide RNA along with Cas9 that was introduced by lentivirus at day 14, and then examined at P21, you can see that there's really beautiful full range of dendritic spines. And you can see that you get all kinds of morphologies. You get nice mushroom guys, you get these beautiful branched guys, and you get those that even form these spinules. Spinules are particularly cool because they're protrusions that are transient off of postsynaptic structures that can penetrate the presynaptic side and release factors into the presynaptic terminal. When we delete formins, you can see that the spine structure is completely abnormal. This is a very, very long spine. You get these incredibly branched spines, and you get spines forming these gigantic spinules. Spine density per micron does not seem to be changed, and the percent of branch spines increases with either of two different guide RNAs, and spines with spinules increase again with either of two independent guide RNAs. So these positive outcomes give us a lot of hope. These are data that we literally got last week. So now we're in a good space in which we can carry out the rest of our screen. So the idea that we have now is that based on formins and different interactions with different cell adhesion proteins, that nucleators work coordinately with cell adhesion molecules to generate what we think of as local signaling environments. And so this is a schematic of a synapse. In the light blue, we have these kind of different nanodomains that are outlined, that are contributed by cell adhesion proteins linked to the actin cytoskeleton, and then engaging a particular set of signaling molecules. This
Anna, can you repeat this?
Yes. Can I repeat it?
Please.
Yeah. So just that I think that synapses are defined or divided into different nanodomains. And so I don't just think this, many people do. And so the data are very good, in fact, that if you look at super resolution microscopy data, that you get these clusters of these nanodomains. What we think is that these nanodomains are built in part by actin polymerization coordinated by adhesion proteins. And they generate these local signaling environments. And the protrusion here is filled with actin. This is a scanning EM taken from Svitkina's lab.
Thanks.
And then, so confirmation of this is come from looking at synapses using STED. And so Abhishek's been doing this in the core using, you have to use a super resolution microscope at Mount Sinai. We have access to a stimulation emission depletion microscope, which can resolve things down to about between 40 and 60 nanometers. So here is a confocal photomicrograph filled with TD tomato, and then immunolabeled for Homer and V-glute. And by STED, you can see that these larger kind of puncta will divide out into smaller nanodomains. That each one of these kinds of sites you can more easily resolve into multiple domains. And so in preliminary analyses of these kinds of micrographs show that, in fact, as you would predict in relatively young neurons, if you incubate them for half an hour with an ARP2/3 inhibitor, you actually lose you increase the percent of spines lacking any Homer puncta. So this would be like eliminating synapses. So this is consistent with our actin work. And as the neurons grow older, they're more resistant to this kind of polymerization. In either 14 days or 21 days, if you inhibit formins, you increase the numbers of spines having only a single nanodomain. You're basically reducing the numbers of multiple domain synapses and increasing the numbers of single domain synapses. So the idea that actin regulates these nanodomains is gaining some credence. These are preliminary data, but we're pretty secure in it. Completely independent evidence comes from some work that we've been doing on a totally different protein, or using a totally different strategy. And so this comes from some work in the lab that we've been doing on a molecule called CIFIP. CIFIP, the relevance here is a part of the WAVE1 complex. So it's very important for recruiting ARP2/3 and generating branched actin filaments in membranes. Significantly, people having reduced levels of CIFIP or copy number variations, either increases or decreases, tends to either tip the balance toward autism or schizophrenia. And several years ago, Joe Buxbaum's group generated a haploinsufficient mouse for CIFIP. We looked at this previously, and we showed that these mice have a protracted period of synapse maturation. And Sylvia de Rubis and independently Joseph Kittler's labs have shown that such mice have dysmorphic spines. And what's been interesting to us is that we see now that we lose SYNGAP from synaptic fractions. Now this sounds kind of random, but I'm going to take you through this to show you what I mean. And so in young neurons, this is the mice lacking CIFIP mature at a more, or synapses from these mice matures more slowly. And so this is just a synaptic fraction. These are the controls, and you can see there's less SYNGAP, there's less PSD95, there's less synaptophysin that fractionate. It's not like they're not there, it's just that they're recruited more poorly, I guess, in these mice. In maturity, though, what we find is we have a sustained loss of SYNGAP while PSD95 and synaptophysin recover. And the significance of the sustained loss of SYNGAP is a synaptic protein that engages temporarily with the scaffold and is thought to regulate long-term potentiation and other forms of plasticity. People having reduced levels of SYNGAP have a syndromic form of intellectual disability, and both in mice lacking a single copy of SYNGAP, just as mice lacking a single copy of SYFIP have a accentuated form of long-term depression that is protein synthesis independent, just like what you see from Fragile X. So these data suggest to us, kind of lend credence to this notion that Actin can help assemble these modules that are going to be, it's almost like a seemingly indirect relationship between SYFIP and SYNGAP, but SYFIP and its coordinated assembly of Actin seems to be essential for actually generating and maintaining SYNGAP as a module, or helping it to transit in and out of the synapse. So I've spoken a lot about what these kinds of modules mean to us on the inside of cells, assembling the actin cytoskeleton, kind of assembling signaling proteins, but I've not talked much about what might be adhering them on the outside. This is actually amongst some of the first studies that we did. This is E-cadherin here and taken from a review by Larry Shapiro, but you can see that cadherins transsynaptically, so this would be extending from another cell, engaged by their EC1 extracellular domains in trans to form a strong calcium-dependent adhesion. They're single pass transmembrane proteins that interact with the cadherin molecules to promote the generation of actin cytoskeleton. We began this work with the late David Coleman when he was at Mount Sinai many years ago, and his postdoc Hide Tanaka. And then it was taken over by Tanya, and then Uslam, and then Martin, and most recently by Roxy. So what we showed many years ago is that cadherins were at the right, and cadherin in particular was at the right place and the right time and at all synapses to be engaging in synapse adhesion. So here's cadherin in these punctate, kind of turquoise puncta here at the edges of a growth cone. It's in the cell body, it's in the axon. Here you see it in patches, and it's also in dendrites. From the very first moments that we can first detect a synapse, we can get a cluster of encadherin. And at least in young neurons, encadherin will cluster at both excitatory and inhibitory, here labeled with GAD synapses. So we see it at both glutamatergic and GABAergic synapses. Additionally, it seems to form the right kind of structure. And so this is again taken from the Shapiro review, but encadherin, we know from some great structural work done from Larry's lab, that they engage in strong adhesion in trans and relatively weak adhesion in cis. But the cis coupled with trans adhesion generates a two-dimensional array structure. And this two-dimensional array is about 262 angstroms apart. That fits in really nicely with the distance across a synaptic cleft, which is between 20 and 30 nanometers. We also have this nice distribution of gold particles along the length of a cleft. So these are serial sections, and you can see a continuation of these gold particles. Here's in another young synapse. So these are P2 hippocampus at postnatal day 2. And these baby synapses have like a nice array of encadherin all along the length of the synapse. And at this stage, or at any stage, the encadherin-based adhesion is completely reliant on the actin cytoskeleton. And so if you block encadherin anytime under a week, you can see that it's a major actin effector at developing synapses. So this is work that Martin did. And if you express in these neurons dominant negative encadherin, and you look for innervation of presynaptic terminals, you can see that in control, you have this lovely cell, and these guys have just these wimpy little tiny terminals. However, if you wait a couple of weeks, the numbers of terminals that they develop are completely similar to what you would see in the control. So whatever they needed at the beginning, they MacGyver their way through another strategy to make synapses late. These data are very strong, either inhibition of cadherins, as I've shown here, or knockdown or knockout prevents synaptogenesis early. Several labs have shown this. Late, there appears to be little apparent interference. It's very similar to what we see with actin polymerization, supporting this notion that at least N-cadherin is a major actin effector at developing synapses. And Takeichi's lab showed that spine shape is similarly disrupted like actin following deletion of this protein that links cadherins to the actin cytoskeleton. So what is cadherin doing at mature synapses? At mature synapses, cadherins are now forming this kind of a spot weld. They're not seen evenly distributed along the length of the synapse. You have N-cadherin is no longer present in these blue GABAergic sites. It's only in excitatory sites. But if you overexpress N-cadherin with a single point mutation that disrupts that trans adhesion between cadherin, N-cadherin trans adhesion, you get these things that look like the dendritic spines you get with actin depolymerization. So at a developing synapse, we have normal cadherin-dependent adhesion throughout the complex. This is a cadherin and actin-dependent structure. This seems to be essential as kind of a scaffold upon which to build the structure following this. So I would argue that if you don't build this in the beginning, that the structure that you form later might work, but it's abnormal. In the more mature site, you still have cadherins, you still have actin, but you free those molecules up to do other things. They seem to be involved in other things instead. So now actin can be engaged to change spine shape without jeopardizing the actual pre to postsynaptic connection. And you can have actin deployed along with cadherins to generate endocytosis, for example. Of course, the synapse itself is filled with other cell adhesion proteins. Cadherins by no means by themselves. And so significantly here we have classic cadherins and beta-1 integrins. Classic cadherins can promote the assembly of synapses or promote the assembly of actin, and it does that via ARP2/3, these branched filaments, whereas integrins can also, depending on who they're interacting with, can also promote ARP2/3 dependent polymerization, but there's several interactions that promote Formin dependent polymerization. And these different CAMs occupy discrete zones. For example, SynCAM is at the edges, and beta-1 integrins form a single cluster that can be almost any point within, whereas neuroligands are more broadly distributed. So what happens when you remove a single transsynaptic module? So these are work that Jessica Nikitschuk did several years ago, and she just did a conditional deletion of N-cadherin using a CAM kinase 2 driver. Deletion begins about P21. You have normal synapse density, normal histological organization, normal spine density distribution, and surprisingly, normal morphology. But what you don't have is LTP or spine expansion. So these were work that Jessica did in collaboration with Chung Zhou when he was here many years ago. And you can see that here in these, this is a neuron that was filled with a fluorophore. And you can see that at baseline, you have this little spine. If you stimulate with a theta burst potential here, you get LTP and spine expansion, and it stays. Whereas in the conditional mutant, this little spine expands and then retracts again. So theta burst potentiation sustained. The conditional mutant, it declines. Spine volume is sustained, and in the conditional mutant, it declines. So consistent with this loss of LTP, she also saw that memory was impaired. She did this in a collaboration with Matt Shapiro's lab using a 6-arm radial maze where animals go in a start arm. They're taught to go to a goal arm on a visible platform, and then the platform is submerged, and it's submerged in a different position each day. And they have to learn where the goal arm is, and then they will continue to go to the goal arm over several trials. All the animals could learn the visible platform task, no problem, but our poor N-cadherin conditional mutant mice were impaired in finding the submerged platform. So they traveled greater distance, they made more errors, and the floxed control mice made more perseverative errors, suggesting that they remembered better where the platform was the day before. And whereas the conditional mutant mice started each day afresh. When we made the task harder by shifting the platform to three different sites within the same session, and a wind shift kind of assay. The conditional mutant mice did worse. And so what happens with that, when you make it more challenging, the control mice get better over the course of days. So day 3, 4, they're decreasing their error rate, whereas the conditional mutant mice just fail to learn. This change in performance is coupled with reductions in other modules. GluA1 is reduced and PSD95 is reduced. This is interesting. This is likely contributing to that defect in LTP. It also leads to a compensatory abnormal change in EI balance. Previous work has shown when you decrease PSD95, you can get a compensatory increase in GABAergic innervation. Shekhar-Patil tested this by injecting mice with canic acid, and he found, in fact, that the conditional mutant mice had a higher threshold to seizure. So this is canic acid injections. The seizure stage is here color-coded, orange being seizure, highest levels of seizure, or red, cooler colors being low levels of seizure. So in saline, they have no seizures at all. And you can see that the conditional mutant mice have fewer seizures and they recover faster, and GAD innervation increases. Cadherin 2 or N-cadherin molecules are not unique. If you do a conditional deletion of beta-1 integrins, you see the same thing. This is work from Ron Davis's lab, where he showed no LTP, no working memory. And we then tested these same mice and found that, in fact, the adhesion molecules were totally reorganized. So in the absence of beta-1 integrins, you have an increase in neuroligands, and N-cadherin is now seen in the lysates as well as in the synaptic fractions. So you get a reorganization of the synaptic adhesion proteins following the deletion of a single synaptic adhesion protein. So in addition to a loss of function, you have to think of a gain of abnormal function. So I've told you that the nucleators appear to target subcellular domains, that removing these actin nucleators seems to generate additional domains, and that removing cell adhesion proteins can alter actin-mediated functions and disrupt associated adhesion complexes. So this is all kind of taken from the notion that you have a single generic adhesion protein, maybe one from each family, and yet you have many, many different versions of them. So why, for example, do you have 100 different cadherins? This, in fact, is something that people have thought about first when cadherins were first discovered. So Takeichi and Steinberg at the time in the 70s and the 80s, they found that cells in the periphery expressing different adhesion proteins would sort from one another in order to generate different tissues. And so different cadherins led to different tissues, or to different cell sorting, which led to different tissues. And that was thought to possibly be operative in the brain. And so this notion of the cadherin code has gained a lot of evidence. Some of the first data were everyone looking at where different cadherins were distributed in the brain. This is taken from Idil Bekharoff in the lab, when she looked at N-cadherin, cadherin 6, 8, 9, and 10. And you can see that each one of these has a different distribution pattern, concentrating in different cellular regions and cellular domains, but also that those patterns are partially overlapping. Single cell RNA-seq, this is just taken from the Allen Brain Atlas, shows the same thing, where you have different cellular groupings here, like layer 4-5 IT cells they don't lack CAD9, they have CAD4, no CAD6, and they do have CAD8. So this kind of sorting helps, or is the idea behind it, is that it can help regulate regional variations. But in truth, this kind of sorting is thought to be more important for synapse selectivity. And so this is data taken from an old paper that George and I did a long time ago, where we saw that N-cadherin is concentrated in the barrel centers. It isn't just concentrated there, but it's concentrated at synapses. If you inject an anterograde tracer into the ventrobasal complex and you look at high magnification, you can see the fluororuby stained terminals co-localized with N-cadherin and PSD 95. So these terminals here are actually synaptic puncta, and he went on with Orlando Gill to show that in fact cadherin 8 is enriched in the posterior medial complex of the thalamus, which runs in concert with the septa enrichment of cadherin 8. And so in the last couple of slides, I'm going to show you how we've been pursuing what cadherin 8 has been doing in the context of synapse selectivity. We have been examining cadherin 8 for several different reasons. So this is a project that has been led by Roxana Macias in the lab. Joe Zaki contributed when he was a rotating student. It was initiated by Lauren when she was in the lab, and Karen and Angelo have also contributed. But the CAD8 interested us for several reasons, one of which is there's a deletion, a very rare deletion that leads to a form of autism and an intellectual disability. But its distribution pattern is absolute. So this is CAD8 driving LAC-Z expression. And so this is the LAC-Z concentration. You can see huge increase or enhancement of expression medially in the frontal cortex of the mouse. And you can see this nice enrichment in the dorsal striatum. This timing is consistent with the innervation and synaptogenesis between these two domains. And so in here you see not just enrichment here, but you see a gradient of high concentrations medially, falling off laterally, and falling off ventrally. It's also expressed at synapses. So CAD8 is enriched at sites having SYNGAP, V-GLUT, and you can see here by immuno-EM, you get a concentration of immunogold particles at the striatum, at synapses within the dorsal striatum. So Roxy has crossed these CAD8 conditional mutant mice with mice expressing an RBP4 CRE, which is expressed in layer 5 neurons, and then crossed them with an AI9 TD tomato reporter. And what you can see is when you delete cadherin 8 from all layer 5 cells, that you in fact, these are the controls. So you can see that normally the layer 5 cells have a nice projection pattern to the dorsal striatum, evenly distributed across much of the dorsal striatum. Whereas in the conditional mutant mouse, what you get is now in the absence of presynaptic CAD8, you have still projections to the striatum. So they can still target the striatum. If anything, there may be a little denser within this zone, but what's clear is that you're missing this periphery and that you have a huge increase in what appears to be callosally directed fibers. And so these data tell us that, in fact, well, other data that she's been working on has indicated that synapse density, in fact, is the same across the region. So we know that this domain is now occupied by something else. If she just focuses on terminals coming from the prefrontal cortex by using AAV GFP injected into ACPL in the frontal medial zone of the mouse cortex, and then she looks at where the terminals land in the striatum. You can see in this map that Joe Zaki developed a way for us to look at the terminals in a way that was easy for us to see and to compare across ages and across different injections. So this is a hotspot analysis, and the hotspot analysis lets you compare what's happening in your neighborhood with what's happening. So it takes the difference of a neighborhood concentration to the entire region. and it assigns a z-score of that neighborhood relative to the entire region. And what you can see here is you have medially a higher concentration dropping off laterally, and it's higher dorsally, lower ventrally. From the frontal cortical injections, you can see here really dramatically that almost everything is concentrated medially, dropping off laterally, and it's also kind of clumpy. These data are interesting because in addition to apparent mistargeting, normally axons kind of tile out from one another. They don't concentrate next to one another, and it's suggesting that in fact they're not tiling out as well as they should, in addition to changes in connectivity. Postsynaptically, if she introduces a short hairpin using in utero electroporation into just the postsynaptic neurons, and this time expressing this as a short hairpin against cadherin 8. She sees an interesting phenomenon where normally you have the neurons occupy almost a spherical space, and the color coding here in shows the depth. So everything that's the same color is occupying the same Z plane. Those things that are different colors are occupying different Z planes. And so you can see here with the cadherin deletion that even within the same Z plane, you have several dendrites contacting one another. This lack of dendrite self-recognition is also consistent with this absence that we're getting in sufficient tiling, and suggests that in fact, rather than a anti-adhesive defect, it's like the lack of cadherin 8 is generating additional adhesion. It's a lack of dendrite self-recognition. The self-crossings go up, and we have some changes in a shoal analysis of the length. So synapse density across the dorsal striatum is apparently normal, suggesting that other connections are coming in and taking that space. And the CAD8 actions in dorsal striatum are suggesting that rather than simple point-to-point specificity, we're getting both cell autonomous and non-cell autonomous effects. And cadherin 8 alone dependent, and the lack of cadherin 8 also seems to be revealing some self-recognition phenotypes. So I've told you several pieces, I've provided for you several pieces of data that show that these nucleators appear to work coordinately with cell adhesion molecules in order to generate local signaling environments. Some of this is still preliminary, but I think that the data are coalescing around this idea. So why does this matter? Our data show that in the absence of such a preferred mechanism, that neurons seem to kind of MacGyver their way through circuit assembly in a process that often generates a gain in abnormal function. This is particularly relevant for the project that I haven't talked about today, which is on a Parkinson's disease model, where I think the entire animal seems to be a gain in abnormal function. And an immediate fix is the idea may, in fact, generate some kind of an improvement, but it also seems to maybe impart some kind of long-term fragility. And so I'd like to thank all the people who have worked on this over the years and currently. So the PD team that I was ignoring, this is them. And so all of the work on both the Synapse side and the Parkinson's disease side is done in collaboration with George Huntley, Chris, Ian, Pam, and Swati, and most recently Mofida. She's now in grad school at Penn State. Thanks for all your work on the PD project, and I'll talk about it next time. And for the Synapse team, this is Roxy, Abhishek, and most recently Fatima and Chanel, who are now both in school. Thank you. And then we have a series of students on both teams, Kumail Alu, Kyomi Blake, Kelly Turner, and Emily Dodd. I need to thank my funders and, of course, collaborators, particularly in this project. It was Mark and Tim and Uslam. And I have to shout out to the Microscopy and Bioimaging Core. This is the entire group, but this is a nicer picture. So this is Shilpa, Glenn, Bill, Esperanza, Allison, Javier, and I can't see Nikos because he's blocked by my view, but Nikos I know is there. And so Allison and Bill helped with some of the EMs, And Esperanza and Shilpa and Nikos have been instrumental in helping Abhishek get this project off the ground. So I will take any questions. Thanks.
Thanks very much, Deanna. That was a great talk, great overview. We do have time for questions. I'd encourage people to unmute themselves and just ask Deanna directly.
Deanna, this is Spiros Efthymiopoulos from Athens.
Yeah. How you doing?
Good, good. How are you?
Good.
I wanted to ask you whether synaptic adherent junctions being around the synapse, whether do they prevent the neurotransmitter of diffusing away and probably reaching the astrocytes, or in a different way, how do they affect the interaction of the synaptic components with the astrocytes, if there is an interaction?
Yeah, I mean, this is something that I like purposely ignored because it's almost too big of a topic. But the astrocytic interactions with synapses, as you know, are among the most dynamic. And so it's one of these, it's constantly changing depending on the environment, and it's constantly changing. I think that while astrocytes are constantly there at the edges of the synapse, that relationship changes in part based on things like MMPs, right? And so you would know better about this than me, I think, but you could have, it seems as though depending on levels of activity, you can have temporary changes between this adhesive environment at the kind of sacrosanct zone in the middle of the synapse and the edges where these astrocytes seem to work. And that edge effect seems to be extremely dynamic. And so I didn't address this at all, but I think that the actin interactions have a lot to do with that.
Okay, thank you very much.
Thanks, Spiros. Other questions? Don't be bashful, everybody. You know, Deanna, I'm struck in listening to your talk, which I found to be an extremely helpful overview, is the sheer number of proteins and the complexity of processes that come together to construct a dendritic spine and control its life cycle. I mean, it really is daunting to, as someone whose lab has tried to understand how cocaine modifies spines, for example. It's really daunting to think about how to even go about that, given the complexity.
But I think it's funny, I think on the one hand it is complex, but on the other hand, I feel like it's, once we master the complexity, we'll see where there are those sensitive points. And I feel like things like there are certain points that are more sensitive than others that are going to be the more treatable sites, or you know what I mean? So I don't think it's as complicated. It won't be as complicated in a disease-related sense as it is in just a biological sense.
Mm-hmm.
I hope.
Yeah, great work. Well, we're at 2 o'clock. So again, thank you so much for a great talk. And thank you everybody for joining us, and see you around campus. Bye-bye.